Hello guys, welcome back to the Gauravyadav channel. In today's lecture, we are going to study about some biochemical tests for the identification of the microorganism. So, in today's lecture, I'm going to tell you about IMViC test. IMViC test is a biochemical test which is usually designed to differentiate the gram-negative intestinal bacilli from the which comes under the family of Enterobacteriaceae, particularly Escherichia coli and the Enterobacter and the Klebsiella group, on the basis of the biochemical properties and the enzymatic reaction in the presence of specific substrate. So IMViC test basically consists of four different type of tests. First is I. I stands for indole production. Second is M. M stands for Methyl Red. V stands for Voges Proskauer. Small I is just for the fluent pronunciation of the test. C stands for the citrate utilization. So these are the four basic tests which comes under the IMViC, which is usually a biochemical test which are used for the identification or the differentiation of the different type of bacteria which comes under the Enterobacteriaceae family, like Escherichia coli, Enterobacter aerogenes and Klebsiella species. So first is indole production test. Indole production test is used to detect the indole production by the microorganism. So what we will do, we will prepare a culture medium, basically a broth medium, like peptone water, which will contain a tryptophan. Tryptophan is an essential amino acid. What we will do, we will inoculate the organism into that medium and leave it for the overnight incubation. After the overnight incubation, we will add a few drops of Indole reagent or Covax reagent, whose scientific name is dimethyl amino benzaldehyde. After the addition of the what happened in that medium, the tryptophan is converted into indole plus pyruvic acid plus ammonia, that is NH3, by the help of tryptophanase enzyme. And after the addition of reagent, and there will be another reaction which is like indole plus Covax reagent, which is dimethyl amino benzaldehyde, which contain HCl. It will lead to formation of rose indole dye plus water. So the positive test is indicated by pink ring or cherry red color. Positive indole gives pink ring and negative indole test gives yellow ring. The E. coli gives indole positive and Klebsiella and Salmonella give indole. This is the picture of indole test. As you can see, there is a formation of cherry red ring. So this is the positive test. In this test there is a yellow color ring. This means this is a negative indole production test. Second is Methyl Red and Voges Proskauer test. Methyl Red and Voges Proskauer test is used to differentiate enteric facultative anaerobes that produce large amount of acid and neutral product, that is acetone, as an end product. Both of these tests perform simultaneously because they are physiologically related and performed in the same area known as MRVP broth. So production of organic acid gives red color on addition of methyl red indicator, that's mean MR positive and VP negative. And production of non-acidic products such as ethanol, acetone gives yellow color, that means negative result. This shows MR negative and VP positive result. How we will do that test? First we will prepare methyl red methyl MRVP medium and a two different test tube, and we will inoculate the organism in both the test tubes and leave it for the overnight incubation. After the overnight incubation, we will add both indicator in their respective test tube. So in MR test tube we will add methyl red indicator. In VP test tube we will add VP indicator, which comes under a VPA reagent and VPB reagent. So this is the result of the MRVP test. As you can see, there is a formation of red color. This is a positive result, and in negative result it will show yellow color. Same as with VP Voges Proskauer test, VP positive shows red color or VP negative shows yellow color. E. coli is MR positive and VP negative, and Enterobacter aerogenes is MR negative and VP positive. So those who produce acid will give MR positive, and those who produce ethanol or acetone, means non-acidic product, will give VP positive and MR negative. Fourth test is citrate utilization. Citrate utilization is used to detect the ability of certain bacteria to utilize citrate as the sole source of the carbon. So citrate utilization test done in a Siemens citrate medium that contains sodium citrate and bromothiamyl blue as in the indicator. Citrate utilization depends upon the enzyme citrase, which is produced by the organism, and that break down the citrate to oxaloacetic acid and acetic acid, which turns a medium into the blue color. So there is when we inoculate the organism in a Siemens citrate medium, there will be, if that organism is positive, there will be a reaction which is as follows. Citric acid is converted to oxaloacetic acid plus acetic acid by the help of the enzyme citrase, and oxaloacetic acid and acetic acid is converted to pyruvic acid plus carbon dioxide by the help of the certain enzymes. So the citrate positive gives blue color, means blue color only form when the medium pH is alkaline, means above the 7.5 pH, and the citrate negative gives green color if the means the pH should be acidic. So the Klebsiella gives positive citrate utilization test and E. coli gives negative citrate utilization test. So there is a dye which is known as bromothiamyl blue dye. So this dye plays a very important role. How? Because this dye in the pH of acidic, in the acidic medium, it will form a green color, but in the alkaline medium it will form a blue color, which gives the positive test. So this is the picture of citrate utilization test. As you can see, organism turns this medium into the blue color, so this means this is a positive result. As you can see, this medium is still green in color, so this indicates a negative citrate utilization test. So these are the IMViC tests which we perform basically in the medical microbiology laboratory to differentiate the microorganism, to find out the species of the organism on the basis of biochemical characterization. So in the next lecture we will again discuss some different type of biochemical test like TSI, nitrate test, urease test, oxidase test, coagulase test. These are also a biochemical test. So I will discuss these tests in the next lecture. So if you didn't subscribe my channel, subscribe it, like my video, share my video, and do not forget to press the bell icon. And thank you for watching my video. Have a nice day.